this idea that I'm explaining to you about the idea of having pathways is not a new idea, nor is it something that's so quirky to me, but actually there's a whole range of people building up resources for pathway biology. And we'll probably have a wee look at some of those in a moment. And if you go to this site here, Path Guide, so this is essentially a website that has been devoted itself to actually listing all the potential resources that you might have for pathway stroke interaction biology, and it lists hundreds of different sites. Okay, some of these ones here are some of the more better known ones, and I'll just quickly go through these now. So KEGG is perhaps one of the best known pathways. Does anyone know KEGG? Never heard of KEGG? Okay, so as these resources go, for those who are in the research environment, KEGG is probably one of the best known resources. So this is from Kyoto in Japan. And essentially this started off as a biochemical resource for biochemical pathways, and over the years they have evolved into different signaling pathways, cancer biology, disease pathways, etc., etc. Again, perhaps I won't go through them during today, but I present the links there, and you can go and have a look at those at your leisure. But essentially what KEGG has done is it's got a notation. So I want to introduce, first of all, the idea of a notation system. So a notation system is a way of drawing, okay? So it's a formalized system by which you can represent entities and interactions between entities. And so if we look over here, so here they show that this line with a plus P, that's phosphorylating this. This is a dephosphorylation because it's a minus P, okay? I can't actually read these, but these are various other interactions, and you can glycosylation, methylation, activation, inhibition, etc., etc. So they use different lines to represent different events, and I think it's reasonable to say that it's a usable system, but it's actually quite a crude system in terms of what you can represent. And you'll notice in here that a lot of these square boxes here, they list an enzyme number. So most enzymes have a number, so that means that actually that enzymatic reaction from here to here can be classified irrespective of what species it is. So in some ways, the nice thing about having a number is it doesn't actually define a specific protein, it defines a reaction step. But of course, mapping your genes onto an EC number is possible, but it's not easy. Now this is something called a molecular interaction map. I find them almost impossible to interpret. So there are many ideas how to do what we are discussing today. This is the one which I find more difficult to understand. So you'll see it's almost drawn like a wiring diagram, and that's kind of what it is. Again, there is a notation system that shows you what is happening here. And actually, the detail of this is very difficult to follow. As I say, I mean, obviously, some people can follow it because they're drawing these diagrams. I just personally find it a difficult thing to do. You will also notice, perhaps, that actually we use labels to define what it is that we're talking about. Quite often, you'll see in these systems that people don't actually use a standard way of describing something. So if I look here, Psych B, which presumably is cyclin B, which if you looked yesterday, the gene symbol for that is CCMB, okay, or B1 or B2. It doesn't say here. And in these cases, they're using non-standard nomenclature to describe entities. So that means instantly you're not entirely sure what it is that's being represented, potentially. Okay, so if you use a symbol which is ambiguous, then you're not sure exactly what is being represented. So we've heard of Wikipedia. So this is a effort to describe pathways on a community-based curation idea. Now, whilst Wikipedia, we all use it, it's fantastic. It's amazing that it works, though, isn't it? The idea that anyone can come down and edit something and actually you get something sensible back. Unfortunately, I would argue that this has not necessarily been achieved in terms of wiki pathways. So again, their notation system, first of all, is very limiting to what you can represent. So if you want to represent a complex, you kind of draw it together like that. And then you've got all of these inhibitions coming in here. And actually, even on this level, which is okay. I think, what are we looking at? So this also looks like a cell cycle. In fact, it is G1S, G2 M phase. I find it difficult to follow. Again, it's yeah. I mean, I think it's very limiting in towards what you can represent. So the other thing is actually quite often is what you end up with that. So this is the most edited pathway on Wikipedia. I checked it out yesterday. I've put this there. I don't think it's been changed. So it's had 400 edits, and what you've got is this completely incomprehensible mishmash of lines, and who knows. So if you can't read something, how do you know if anything is right? You've got to be able to visually check the information, and that is just a mess. So that's potentially one of the downsides of letting people do your editing in a pathway sense. So this is another one. Again, this is run by Nature, or has a link to Nature, Pathway Interaction Database. Again, you can see this is a complex, and they list the proteins in that complex. Again, the notation system for describing interactions is very crude. Okay. And perhaps one of the best, but still limited, is reactome. So this is coming from, I think, labs in Canada and EBI. It's worth actually having a look at that. This is, again, quite an extensive now series of pathways which have been curated. So their model for this is actually they have a team of curators sitting in Canada and Cambridge who sit down. First of all, they decide what it is that they're going to model. Then they bring in experts. Then they try and have this dialogue with those experts and produce a model of what they are. But you can't actually edit this. It's something, well, you can't edit it, and those edits arrive here. But it does describe quite a lot of things. And it is also a language which is very similar to the one I'm going to describe today. It is based on this idea of a process diagram. And we'll describe a little bit more about the process diagram in a moment.